you going to do an introduction or? All right. I mean, whatever you want. Just. I mean, you also said 3 after, and according to my clock, it's only 1 after, so. To remember who the heck I am. Who is this person?
Right. I don't know.
She wandered in and said she was giving a seminar, so we just like set up the room real quick. We didn't want to say no.
Right.
She dressed the part. She looked like she was going to give a seminar. Yes. Did I not email everyone? Did I only email whoever suggested it, Bradley? I said yes. John said yes. And then Williaming. Oh, I also know that. I could, as an undergrad. Yeah, I did my undergrad at Madison.
Still crazy.
All right, thank you all for coming. We're thrilled to have Dr. Amanda Amadeo here today. Dr. Amadeo trained in various organisms across all of biology at the University of Wisconsin-Madison, and then got her postdoc at Stanford with Jens Spacetime and Aaron Strait. She's now a Sigler Fellow at Princeton, and so very much looking forward to hearing about the brilliant work she's been doing.
Thank you. Okay, so first, interrupt and stop me if I say something you don't understand or if you have questions, because if you're lost, someone else is probably also lost, and I would much rather be stopped and explain something before I give the whole talk and you had no idea what I was talking about. So I'm going to try to give you all the information you need, but if there's something on a slide you don't get, let me know. Okay, so let's start with the very simple observation that all cell types have a characteristic size. So muscle cells are super big, and they need to be big because they need to generate the forces required to contract your muscles. And red blood cells are really tiny, and they need to be really tiny because they need to squeeze through capillaries in order to oxygenate your tissues, right? Okay, that's an obvious fact. One thing that all of these, that's well conserved across all of these length scales, though, is the nuclear to cytoplasmic ratio. So really big cells, like muscle cells, are able to get as big as they get because either they do what muscle cells do, which is have many nuclei, or they undergo endocycles and they have one really giant nucleus. But in general, cells that are sort of larger than the typical cell size for the organism, which is represented here by these skin cells in the middle, are multinucleate. And then red blood cells are sort of cheating because they actually eject their nuclei in order to get quite as tiny as they get. And so this nuclear to cytoplasmic ratio, or NC ratio, is conserved not just within species, but across species. So the genome size is predictive of cell size, here represented by diameter of red blood cells, across many, many species of mammals. And in fact, it is so well conserved that deviations from typical nuclear cytoplasmic ratios are used as diagnostic criterion for several types of cancers. So for these urothelial carcinoma cells, anything below about a 0.5 is considered sort of normal morphology. Between a 0.5 and a 0.7 is considered cause for concern, but not necessarily diagnostic. And anything above a 0.7 is actually diagnostic and associated with poor prognosis in these cancer types. So as with anything in biology, if there's a rule, there's an exception. And one good exception to this rule is the early embryo. And this is because eggs are huge cells. They're the largest cells even in mammalian bodies. They're the largest cells. And especially for those of the eggs of large externally fertilized species, they need to be huge because they need to contain all of the material to make whatever hatches out of that egg, whatever larval form, a tadpole or maggot or whatever comes out of that egg. So the very first order of business for a newly fertilized egg is actually to restore a more typical nuclear to cytoplasmic ratio. And they do this by undergoing a series of cleavage divisions. And in these large externally fertilized eggs, these cleavage divisions are actually somewhat controlled by the NC ratio, and I'll get to that in a second. So one of these large externally fertilized eggs that I've been telling you about is this Xenopus frog egg. And you can see this is sped up. They don't go quite this fast, but they do incredibly rapid divisions during their early cell cycles. Their cell cycles are about 30 minutes, and they're able to do this because they omit both of the growth phases, which of course they do because these divisions are reductive. These divisions are entirely driven by maternal components and therefore are synchronous because the cells really don't know yet what they're going to be. They haven't really started to differentiate yet. During these divisions, they're just little dividing machines. The cells are non-motile. They just divide, divide, divide. And there's very little zygotic transcription. So really all they're doing is dividing. They're not transcribing. They're just using maternally provided RNAs and proteins. And all of this changes at a time called the mid-blastula transition, or MBT. So at the MBT, the cell cycle slows with the addition of G2. You start to get patterned divisions. So now cells are going to start dividing depending on their position in the embryo. Right after this mid-blastula transition, you get the onset of gastrulation, so tissues are going to start folding over themselves and doing major morphogenic movements. And very importantly for my talk, you get the activation of the zygotic genome. So now the phenotype of the embryo is dependent on the zygotic genome rather than what the mother was. And as I kind of alluded to earlier, this is actually not only a process that restores the NC ratio, but it's actually controlled by the NC ratio, at least in part. So if you alter the NC ratio, you can alter the number of divisions that the embryo does before stopping. So this is really elegantly shown by this experiment from Newport and Kirshner in 1982. This is like one of my favorite experiments. What they did was they took an embryo, they fertilized it, and then they tied the embryo in half with a baby hair so that all of the DNA is on one half of the embryo, and so you have this anucleate side of the embryo and then sort of a normally dividing embryo. They then allowed that thing to develop and divide for a little bit. And then they removed the ligature, like they removed the hair, and they allowed some of the DNA to slip back through. So now you end up with this situation where you have an embryo whose cytoplasm was all fertilized at the same time, whose nuclei have all undergone the same number of divisions, but where the nuclear cytoplasmic ratio is very different on the two halves of the embryo. And so they just watched these things develop. And what they saw is that on the half of the embryo where the nuclear cytoplasmic ratio is increased, this half stops earlier after fewer numbers of divisions than the half with the decreased nuclear cytoplasmic ratio. Really nicely illustrating that this ratio is in control of the number of divisions that the embryo undergoes before cell cycle slowing. And you can also alter this ratio in basically any way you choose. So you can change the ploidy of the embryo, which changes the amount of DNA, of course. If you make a haploid, they'll do extra divisions. If you make a tetraploid, they'll do fewer divisions. You can suck out cytoplasm. Whatever you want to do, the embryo will respond accordingly to the nuclear cytoplasmic ratio to restore the correct cell size. OK, so then the obvious question, and the question that's been in the field for quite some time, is how is the nuclear cytoplasmic ratio sensed? And sort of, yeah, you guys want to come in? Yeah, there's some chairs. So how is this ratio sensed? The textbook answer, if you go to like any classic developmental biology textbook, you will see this hypothesis that the mother loads some component, this purple stuff, into the embryo, and it loads a set amount of that, so this stuff is relatively constant. Then the nucleus takes up some of that, but there's not very many nuclei at the start. They divide, they take up more, they divide, they take up more. Eventually, the number of nuclei actually starts to make a difference in the amount of this stuff. At some point, you run out, and then you get cell cycle slowing and transcriptional activation. This is the idea. It's called the titratable inhibitor hypothesis, and it's been the state of the field for about a little less than, since the Newport and Kirshner paper, so 82, a little less than 40 years. And this is sort of where I came in. So you get cell cycle slowing, you get transcriptional activation. This is sort of where I came in. When the we know that when the nuclear cytoplasmic ratio is low, we have activating cues to the cell cycle. This is better established, so I'm drawing it as a solid arrow. And then we know that transcription is inhibited. And the reason I draw this arrow as dashed is because we also know that there are feedbacks between these two things. And so the inhibition of transcription could, in theory, be indirect. So we'll get into more detail in this later in the talk, but we know that in Drosophila, at least, certain transcripts that are cell cycle inhibitors are some of the earliest transcripts, so we know that this arrow goes this way. And also, it's hypothesized in the field that the active transcription itself may lead to replication stress that may be causal in cell cycle slowing. And in this direction, it's sort of necessarily true that if the cell cycles are very, very fast, you just don't have that much time to transcribe. So there are these two arrows. So during my PhD work, we decided to do some really, really naive experiments, which is why it's good to have naive PhD students who don't know any better. So what I did, and this should not have worked, what I did was to take purified cytoplasm, which is easily obtainable in Xenopus. It's called Xenopus egg extract, and so you can just centrifuge Xenopus eggs and get cytoplasm from them. And then take DNA from the sperm of Xenopus males, and then add it at different concentrations into this extract and ask, by the addition of radiolabeled UTP, if we can get NC ratio dependent transcription in a test tube, right? Really naive experiment.
But what is NC ratio in this case? Just concentrations?
Concentration of DNA. So yeah. Yeah, so I use the term NC ratio and DNA ratio, DNA cytoplasmic ratio, interchangeably. I think the ploidy experiments, so when you change the ploidy, right, you make a haploid embryo, that cell is going to have the same number of nuclei, but each nucleus has half the amount of DNA. Right. And so in the literature, it's called NC ratio, which is why I say NC ratio. But you can think the N component of this is pretty well established to be DNA.
Yeah, okay. So we should think about it exactly like DNA to solve cytoplasmic solution, whatever
yes.
it is.
Yeah. I think that's the way I think about it for sure, and I think that's the way.
And cytoplasm, of course, contains all proteins or whatever, so it's a solution.
Yeah, and there's like a lot of stuff inside of right, and so
metabolized proteins. So.
Yeah, and in theory, that purple thing, the inhibitor, the titrated inhibitor, could be any of those, right? And so this is you're anticipating where I'm going. So what we saw when we put different concentrations of DNA into extract was that indeed we could get NC ratio dependent transcription. So below about 50 nanograms of DNA per microliter extract, there's no transcription. The gray stuff on the gel is the radiolabeled UTP transcription. And above that, we do get transcription. And this threshold, and it's very nice and threshold-like, this threshold happens almost exactly where that concentration is in the embryo when it stops dividing. So that was really gratifying. I want to point out, like this, we didn't completely recapitulate the mid-blastular transition in a test tube. This is only a very small number of transcripts. There's really one major band here, which is a POLT3 transcript. But this gives us...
ATP is already there in the cytoplasm, right? So you don't have a flux of ATP or
so, yeah, when we make the Xenopus egg extract, we actually add to it an ATP regenerating system so that keeps it alive and healthy. So ATP is being regenerated in this, and ATP would be a dangerous thing to run out of too. So, but ATP is not is not the thing here. Okay, cool. So we didn't completely recapitulate the MBT, but we do have a really good system where we can now biochemically purify and track this activity. And so this was all published. This is my graduate work. This has all been published for quite some time. So I won't take you through the gory details, but I spent some time in the cold room, did a biochemical purification, and identified the thing that is inhibiting transcription in the egg extract below the threshold concentration in order to identify histones as the titrated inhibitor. And so histones make a really good candidate for the titrated inhibitor. In particular, histones H3 and H4 are the ones that came out of my, my purification. They make a really great candidate for the titrated inhibitor because they're super abundant. They're the most abundant chromatin protein. They are the thing that wraps DNA into nucleosomes, these beads on a string. And so they directly bind DNA, so it's very easy to imagine how they would be titrated against DNA. And it was already known in the literature, well established, that nucleosomes sitting at transcriptional start sites, sitting at the place where transcription begins, actually are inhibitory toward RNA polymerase binding those transcriptional start sites. So you could imagine, it immediately sort of a lot of ideas about how this might work, right? And they make a really good candidate. Okay, so let's add this to our model. We got histones are inhibiting transcription. So we're going to add histones, and then we're going to strengthen this arrow because that was direct in the extract. There were no cell cycles in this extract. All right, so at this point, I left my PhD and left frogs and moved into Drosophila. And I moved into Drosophila because I really wanted to take advantage of their wonderful genetics, genomics. They have a very small, well-annotated genome, especially compared to Xenopus, and especially the quantitative imaging. This is actually not quantitative imaging, but it's really pretty, available in Drosophila. So Drosophila MBT is slightly different than Xenopus MBT in that they do one additional division compared to Xenopus, so they do 13 fast divisions, and they have even faster cell cycles because they omit cytokinesis. So for the first 13 divisions, all of these divisions are done in a common cytoplasm, and then when they stop dividing in cycle 14, then all the membranes come down at once to cellularize all of these nuclei at the surface all at the same time. So that's a little quirk of Drosophila development, and it means that we call these early cell cycles nuclear cycles, or NCs, which I didn't make the nomenclature, but so if you see NC in my talk without a slash, I mean nuclear cycle. If it has a slash, I mean nuclear to cytoplasmic ratio. I tried to stay away from the NC nomenclature as much as I could. Okay, so the first thing we wanted to know is this generalizable? Does this work the same way in Drosophila as we think it is in Xenopus? So we wanted to manipulate histone levels and look and see if we got effects on either transcription or cell cycle in flies. So I told you that flies do 13 fast divisions. They're actually not all exactly the same length. The 13th one is a bit longer than the ones previous. And then they slow down in cycle 14. And actually what I'm plotting here, because they don't do a synchronous 14th division, is the amount of time between entry into 14 and the beginning of gastral movements. So this is not real cell cycle time. Okay, so these are the wild type divisions. What happens if we add extra histones? So the prediction is if we add extra of the thing that is inhibiting slowing, we should go longer before we slow. So we should do extra divisions, right? Okay. We do. I wouldn't have shown you the data if we didn't. So the divisions are faster. The early divisions are faster. And then about 10% of these embryos go on to do extra divisions. And they do all kinds of things after they do that extra divisions. They are not happy. Each one dies in its own unique way.
And each dot is an embryo.
Each dot is an embryo. Yeah. Each dot is an embryo. But the majority of these guys, when we overexpress histones, are actually wild type. And we're not really sure why that is. It probably is due to the penetrance of the mutant. It turns out that these flies are very sick. They don't like overexpressing histones.
What is OE?
OE is overexpression.
Overexpression.
Yeah, sorry. It just didn't fit. Yeah, so.
But OE, overexpression, normal is?
They have normal cell cycles. And then a group of them have shortened cell cycles and then extra divisions. And we also.
But can you measure in the two classes and show that the ones that do the extra cycles have cytologically more histones than the ones that are?
So what I'm not telling you is that the vast majority of these embryos are like completely dead and unanalyzable. So when we do big collections, it's tricky. And we only know they're going to do extra divisions when they do extra divisions. And then if they do start making zygotic histones at that time. So this is not like a totally impossible experiment, but it's a difficult experiment and one we haven't done. And if we didn't see a difference, we wouldn't know if it was because we wouldn't know what to control to compare to these guys in cycle 14, or where they will be starting to make their own histones, or these guys earlier, but then we'll be including these guys in that group, right? So it's a little tricky. It's not impossible, and it's a good experiment.
But if you, for control, if you overexpress some negatively charged protein, but not histone.
Histones are positively charged.
I'm sorry. I mean, some positively charged protein which is not histone.
We never tried that. That's a good experiment. Yeah, we never tried that. What I can show you is that it works both ways. So yeah, that's a good experiment. I don't think we're like, the reason that they are sick, like that the embryos are sick, may be just because of the charge balance, because we are definitely overexpressing a very positively charged protein. And in Xenopus, when I inject histones into Xenopus, a lot of them get weird spindle defects and stuff because the histones just gone.
To lots of
Yeah.
stuff in the cell because of
Right.
the
Yeah, you're 100% right. So I wanted to show you this data because it's actually very hard to get extra divisions. There's very few mutants that will do it, and so we were very excited to see this. But we actually focused more on the histone knockdown, which is this experiment. So we can also do the other direction, which is to knock down histones. So this is just wild type again, the controls for this experiment. And when we knock down histones, now about a little less than half the embryos do what this guy does, what these guys do, which is to stop early. And these are actually pretty healthy. They stop early, they begin undergoing gastrulation. They do not hatch, but they die quite late in development. So they get through the early stages of development quite well. And so these are the ones we actually focused on more to fully characterize. And this is an RNAi, so some of them are still relatively wild type. And we here are more confident that that these guys are just not very well knocked down, because we actually did RNA-seq on these guys, and the thing that we were RNAing against is not well knocked down in the ones that do normal cell cycles. But even these ones have somewhat elongated 13s and somewhat shortened 14s. And since this time in 14 is the time until gastrulation, and gastrulation requires transcription, like you need to have zygotic transcripts in order to get gastral movements, we wondered whether or not transcription was affected in these SLBP RNAi embryos. So to look at this, we just did RNA-seq on embryos that were stopped in 13 compared to wild-type control embryos. And what we saw is that a lot of genes are both over and underexpressed. So we get about 3,000 genes that are overexpressed, about 2,000 genes that are underexpressed. So the prediction here is that if we were initiating zygotic transcription early, we should see a lot of genes overexpressed, and these 2,937 genes are indeed enriched for known zygotic transcripts from other experiments. But what about these guys? So it turns out that in order to clear the way for the zygotic transcripts, a whole bunch of maternal transcripts are degraded at the same time. And these yellow underexpressed transcripts are enriched for the maternal transcripts. So it really looks like zygotic genome activation and all of the things that go with it is happening early in these embryos. Okay, but what about these feedbacks between transcription and the cell cycle, right? I told you I was not worried, but I told you that there's a feedback in terms of duration. So when we did these experiments, we were taking these red embryos that are arrested in 13 because we wanted the most affected embryos, and we were taking them, we wanted to make sure that they weren't going to do the wild type behavior, so we were taking them between 30 and 45 minutes into cycle 13, and we're comparing them to wild type embryos at the end of 13, but this should be obvious, if you transcribe for longer, you will have more transcripts, right? Like if you integrate under a larger curve, a longer time curve, you will have more transcripts. So in order to control for this, what we did was to continue to take the same set of embryos arrested in cycle 13, but now compare them to wild type embryos that had made it into 14, so that the total amount of interphase time was matched. So the total amount of time for transcription is matched. This means that these guys have more nuclei So this is a somewhat conserved, right? These guys have more because they've undergone an extra division, or a normal division, but one more compared to our histone knockdowns. So they will have more DNA, so they will have more template for transcription. So this is a relatively conservative estimate of the genes. And we indeed lose about 2/3 of our overexpressed genes and more than that of our underexpressed genes. This makes sense, the underexpressed ones, because they are known to be on a timer since fertilization. We know that a certain product is translated, and that's the thing that is responsible for degradation. So actually, I'm surprised that any of these survived. But the overexpressed genes, we lost about 2/3, which indicates that this feedback between cell cycle duration and, or between transcription and cell cycle duration is really important in this process. There was one thing that jumped out in this overexpressed gene category that I just want to highlight for you, which is a gene called Frühstart, which is a negative cell cycle regulator. It's actually a direct CDK inhibitor. So this gene seems to be quite sensitive to changes in histone levels. It's one of these guys up here. And it is overexpressed even in our more carefully time-controlled experiments. So I'm going to add a gene list. Now we have a list of about 1,000 genes that we think are probably directly sensitive to histone levels. And I'm going to add the cell cycle regulator Frühstart to the genes that are sensitive to the NC ratio by way of histones. So we wanted to investigate the cell cycle duration feedback in more detail. And we wanted to do that in particular because a lot of the ways that people have looked at what genes are or are not sensitive to the nuclear cytoplasmic ratio is by comparing wild-type embryos to haploid embryos. So haploid embryos have this reduced NC ratio. They do one extra division, so they stop in nuclear cycle 15 rather than stopping in 14, and their transcription is delayed. But they also, all of the cell cycles are correspondingly changed in length. And so if you're comparing a nuclear cycle 13 wild type to a nuclear cycle 13 haploid, the haploid is just much younger in total cell cycle time. So it's sort of the reverse of the problem that I just described to you. There's been less time to integrate. So to look at this, we wanted to really look at the dynamics of this transcription. So this is hard. This is hard to assay by sequencing because these cell cycles are, about 10 minutes long, and so getting tight sampling across that duration is actually technically quite difficult, especially because you can't do in vitro fertilization in Drosophila. So you have to manually watch each embryo individually to know how old it is. So we turn to this MS2 MCP system, which allows us to quantitatively image individual transcripts at a time. So the way that this works is that you incorporate 24 of these MS2 stem loops downstream of the cis-regulatory regions of your gene of interest. And then when they are transcribed, they are bound by a maternally provided MCP GFP. And that makes these bright spots in the nucleus whose intensity has been well characterized to be directly proportional to the amount of mRNA being transcribed at that locus at that time. So this system is not high throughput, but it gives us the fine temporal resolution that we need to be able to tell if the gene is responding directly to the nuclear cytoplasmic ratio or indirectly through cell cycle duration. It also gives us subnuclear resolution, so a lot of the interesting early genes are spatially patterned. They're going to give rise to the different segments, body segments of the Drosophila. And so here we can actually see, we're not grinding up the whole embryo, we can actually see where the transcription is happening and if it's normal or perturbed. And then it allows us to control template independent of ploidy. So this is another problem with most of the ways that people have done RNA-seq, which is if you make a haploid, you have half the amount of DNA that can be transcribed. And then you compare that, and then you assume either that it should be double in the diploid, but that's not always true. So here we can circumvent all of that normalization issue. And in the haploid, we have one copy of this MS2. In the diploid, since it's an exogenously provided construct, we can also just put one copy in. And we don't have to do any kind of normalization. OK, So this is one way of plotting this data. We looked at it in many ways. I will spare you all of the details. These are just the average trajectories for all of the nuclei that are transcribing, anywhere we can detect a dot in the entire embryo, for that gene Frühstart that I told you about. And this gene is pretty typical of the 5 or so genes that we looked at. And these are the trajectories for wild type. So it goes up, mitosis happens, it goes down. It goes up in the next cell cycle, mitosis happens, it goes down, etc. And when we make haploids, of course, the cell cycle duration is shorter, but not much else changes. There might be a little difference here, but we're not real convinced that that's real or biologically important. And this was true for pretty much all of the genes that we looked at. There's not a big difference in terms of the kinetics of activation between haploids and diploids. But this is just plotting those nuclei that do activate. So Frühstart is a really nice gene for these sorts of studies because it's incredibly switch-like. The gray bars are all nuclei within the embryo, and the blue are the ones that are transcribing, that we can detect a dot at any point during the cell cycle. And you can see that Frühstart is off until cycle 13, and then it's a knot pattern gene, so basically all of the nuclei pretty much come on at the same time. In the haploid, that switch is delayed. So now they stay off, majority off, in cycle 13 and don't come on until cycle 14. And just to make sure that this isn't some weird downstream consequence of cell cycle duration, right? Like maybe Frühstart comes on late in the cell, that's not true, but maybe something about cell cycle duration is the important thing here. We utilized a mutant in the pathway responsible for cell cycle slowing called CHEK1. So this is a diploid whose cell cycle duration is identical to the haploid. And here, you can see in the diploid with the short cell cycle, it activates just like it would in wild type and not like it did in the haploid. So this is direct NC ratio dependent transcription. And this is a thing that some people in the field don't believe is true, but we've, I think, pretty clear, like, not that they don't believe our data, they don't believe that direct NC ratio dependent transcription is possible, but here we've pretty directly shown that this gene, Frühstart, which is a cell cycle regulator, is responding directly to the nuclear cytoplasmic ratio. So we'll add a direct arrow between the NC ratio and Frühstart. This is not an effect of cell cycle time. It is a direct effect of histones. Or, well, of NC ratio, and it's also directly responsive to histones. Okay. So at this point, yeah. Frühstarts? So Frühstart mutants about 10% show 5% show cell cycle defects, so where they keep trying to go. So this is not the only thing involved in cell cycle slowing. It's sort of a minor feedback, maybe, but it is involved in cell cycle slowing. And so we think maybe on the transcription side, I'll show you more data. I have another model for how I think the cell cycle is actually being slowed. But this gene and another gene called tribbles, which also gives a few % defect in cell cycle slowing, we think are some of the feedbacks to ensure good cell cycle slowing. But I don't think that the major thing that's stopping the cell cycle. I think the major thing that's stopping the cell cycle is that CHEK1 mutant that I used to make the diploids that cycled quickly. And Frühstart is not known to interact with CHEK1. It directly interacts with cyclin.
So what you are, this model kind of, what I don't understand, is it specific kind of this NC dependence of transcription? Is it specific to Frühstart, which is of course cell cycle protein, or what you are trying to say that any protein would respond to?
No.
To that? This is a feedback due to cell cycle regulation, or just some physical properties of chromatin which give rise to high or lower transcription?
So the majority of genes that we looked at did not respond to the NC ratio. So I think Frühstart is relatively special. The MS2 system is not a high throughput system, so we've looked at only about 5 genes. And Frühstart is the best one of those. 3 of them I can confidently say definitely don't, and 1 I'm on the fence about. I think that this is a subset of genes. I'm very interested in understanding what underlies NC ratio sensitivity in this subset of genes. We only identified this year, so we haven't had time to figure out what the thing is. But yeah, I think, right, like what makes a gene NC ratio sensitive or not is a really burning question for me right now. Are there other questions before I move on? Okay. So at this point, we were thinking about, okay, we know histones are affecting transcription. How are histones affecting transcription, right? Like what is going on with the histones? What is going on with the chromatin? So we decided that we should just look and see what was going on with the histones, because if you can watch something, I find that you can often figure out, you often generate insights by just watching a process. So in order to look at this, we tagged histone H3, which we picked because it was the best in the biochemical extracts from frogs, with the green to red photoswitchable fluorophore dendra in the context of a pseudo-endogenous cluster. So the histone genes, the replication-dependent histone genes, the ones that make up the bulk of the chromatin, are in these repetitive clusters with all of their promoters and UTRs. And in wild-type Drosophila, in a single genome, there's about 100 of these back to back to back in one cluster. And so we did not replace that. We just added a 101st cluster on a different chromosome so that we could watch what was going on with histone H3 in early fly development. And what we saw was that it works. We can express it. It goes into the nuclei. It goes onto chromatin. You can switch it from green to red. And so the first thing that we wanted to know is the amount of histone in the nucleus constant, right? I'm proposing this model in which there's a superabundance early, and then it's being titrated out. So is it constant over the early cell cycles? And what we saw was that no, it's not. It's not at all constant. It goes down by about 60% between cycle 10, which is the earliest cycle we can image. Before that, the nuclei are too deep, until cycle 13, which is the last fast cell cycle. Okay, so what's going on? Where does that H3 go? We reasoned that one of two things had to be happening. Either in the early nuclei, there's a bunch of histone that's not bound to DNA that is being lost as more and more nuclei are generated and more of the histone is on DNA. Or in the early cycles, there's a lot more histone on DNA than in the later cycles, like a lot more. And these two things are not mutually exclusive. So to look at the first possibility, what we did was to take advantage of this green to red photoswitching property of the histone dendra to do the following experiment. We switched the nucleus during interphase, then let it divide and look at how much histone made it through into the following interphase. The logic being that if all of the histone in the early nucleus were bound to DNA, then when nuclear envelope breakdown happened, that histone would remain associated with chromatin during mitosis and then get divided into the two daughter nuclei. And the sum of the signal in the two daughter nuclei would equal the original mother nucleus. However, if there was non-DNA bound histone in the nucleus, when nuclear envelope breakdown happened, now these histones would be free to diffuse away, and then less than 100% would make it into the daughter nuclei. Does that logic make sense? All right, so this is what actual data from an experiment like that looks like. So this is cycle 11. We switch. It's constant. So that tells us actually that there's no nuclear export of this protein. Then nuclear envelope breakdown happens right here. You lose a large amount of histone, so that's this case. This delta is the amount of free histone. And then this is mitosis. And they undergo mitosis and then form two daughter nuclei. And these two daughter nuclei together have the amount that was in mitosis, right? The chromosomes separate and half goes to each daughter. So this delta right here is the amount of non-DNA bound histone. And you can see here that this is about 55% of the histone in nuclear cycle 11 was not, in this particular replicate of nuclear cycle 11, was not bound to DNA. So then we can just do this experiment over and over again for the duration of the different cell cycles. And what we found was that in cycle 11, we reach a maximum of about 60% of all of the H3 in the nucleus is not bound to DNA. But by cycle 13, that's down to less than 20% of all of the histone not, in the nucleus is not bound to DNA. So there is this dramatic loss. So we're in this case. There is this dramatic loss of non-DNA bound histones from the nuclei as the number of nuclei increase. And we also investigated this possibility, and I'm not going to show you the data for it. I just want to mark it because it's something I want to talk about in my future directions. But we also found that this is happening too, where you are losing H3 from chromatin, but it is immediately replaced by a variant histone, a non-replication dependent histone called H3.3. So you're losing the bulk H3 and getting a specialist H3 on chromatin in quantities that we can detect through microscopy, so pretty large quantities at this time. Yeah?
Do you think that the excess nuclear H3 has anything to do, earlier we were talking about histones in the nuclear cycle ratio, so like is that part of the reason it's inhibiting, is you have so much extra?
Yes, I do. I do think that.
Cool. On it.
So, yes, that's where I'm going.
Okay.
So, no, like, thank you for the setup. Yes, so we saw this, and this is a great example of where, like, conceptually I had predicted this, right? I believed that there would be non-DNA bound histones in the nucleus from our model. But when we actually physically saw how much histone there wasn't bound to DNA, we got to asking ourselves, could these non-DNA bound histones have a function, or are they just there, right? And so we wanted to ask, is there a function for histones off of DNA? And that's sort of a little heterodox. But could we make a histone that didn't bind DNA? And what would that do? Would that have an effect on transcription or the cell cycle? So what is a histone? A histone is mostly a DNA binding molecule. The vast majority of the molecule is positively charged, folded in these histone folds that then bind DNA. But there is a bit of the histone that's not dedicated to DNA binding, which are the tails. This is, if you've heard about epigenetic chromatin modifications like H3K9, H3K27, H3 letter, anything less than 47. These are in these tails, which are these unstructured domains that kind of flop out outside of the histones. So we thought, if there's going to be regulation off of chromatin, that's probably the place where it's going to be. This was, a wild guess, but it seemed right. So what we did was just cut them off of H3 and H4, because those are our favorites, and fuse them to GFP, and then express those at high levels during oogenesis to put them into the embryo.
So you express just tails, right?
Fused to
Tails
GFP.
fused to GFP,
Yep,
of course.
yep,
but-
tails.
okay.
Just this 47 amino acids and this 34 amino acids of H3 and H4
Which
respectively.
are obviously disordered, right?
They are
Unstructured.
disordered, they're unstructured tails. And they're pretty small.
Are they charged?
They have some lysines in them, yeah. They have it, yeah. They're a little charged. But they're also not that big, so they can't carry that much charge. So, okay, these are the cell cycles plotted again. I'm not plotting the time till gastrulation because Yuki didn't want to make the movies that long. So we're just plotting the cell cycle durations. So these are the same as you've seen before, 11, 12, 13. 13 is a bit longer. Okay, we overexpress the H4 tail, nothing happens. But we overexpress the H3 tail, and now the cell cycles of cycle 13 and even 12 are shortened, and we get about 20% of the embryos doing extra divisions. So this is like we've added, this is actually better than when we overexpressed all of the histones, and this is like we've added more of the inhibitor of transcription or the activator of the cell cycle, but it's only 47 amino acids. Okay, so how could this work? What's special about these 47 amino acids? Well, we recognize that in the human literature, and fortunately histones are almost perfectly conserved from yeast to humans, threonine 11 is actually a target, a phosphosite, of that DNA damage checkpoint check 1 that I used earlier to make the cell cycles go faster. And it's well established in Drosophila that check 1 phosphorylation of CDC25 is the thing that stops the cell cycle. So CDC25 is a cell cycle activator, check 1 phosphorylates it and inactivates it, and T11 on H3 is a check 1 target. Okay, so what if there were, so by mass spec we know that there are between 2 and 3 orders of magnitude more H3 than there is CDC25 in the cell. So what if H3 were competing with CDC25 for check1 activity. So this is what this looks like. So we did this in a test tube. This is what this looks like in a test tube. So these are just phos-tag gels that allow us to visualize gel shifts. So what we did was mix CDC25 with check1 in a test tube, and without ATP, it doesn't phosphorylate. With ATP, it does. Good. That had to work. If we add some random protein that's not supposed to do anything, it doesn't do anything. Good. And if we add different concentrations of histone H3, now at high concentrations, physiological ratios are somewhere in here. At high concentrations, H3 is inhibitory of this phosphorylation, and at lower concentrations, not. So this looks like it could be a competitive inhibitor.
But an obvious experiment, which I'm sure you've done, is to mutate this T11.
We'll get to it. Yes. So before I tell you about the mutation of the T11, we also took advantage of this check1 biosensor that Stefano D'Italia's lab at Duke had made, which is just a chunk of human CDC25 that is phosphorylated by check1 and then exported from the nucleus when it's phosphorylated. And so its kinetics look like this, where in cycle 12, it goes up a little bit, and then in cycle 13, it's really going up a lot. And when we overexpress the GFP H3 tail, now this is just constitutively less active. It still does go up, and not all of these embryos stop early, but it's just constitutively dampened down. We also did the experiment that you asked for, which is in the context of this H3 tail, we mutated this T11. So just to remind you, wild type, the full length tail, like the wild type tail, T11 mutant perfectly restores back to wild type cell cycle times. So it's this one amino acid on this tail is responsible for this entire phenotype. And just, if you're a cell cycle aficionado, you might be thinking, well, T11's really close to, right next door to S10, which is another known phosphosite on histone H3. It's an Aurora B site, and people use it as a marker for mitosis. So maybe, when you mutate T11, you're screwing up Aurora B interaction with the histone tail, and it's really Aurora B and not check1 at all. So we mutated the 2 Aurora B sites in the H3 tail as well, which is S10 and S28, and those still have the shortened cell cycle time. So it's not the Aurora, it's the check1 site. One more experiment. We wanted to go the other way, right? So this is all in the context of overexpression of the tail. What if we were to mutate this residue in the context of the wild type? So it turns out that this Zhenbao Dai's lab in China had undertaken this heroic effort just 1 year before we needed them, so good timing, where they had gone through and replaced all 100 copies of the endogenous locus with 10 copies of the histone cluster, in which they systematically mutated every residue that there was any report in the literature, one by one, of any modification. So all of the lysines, all of the serines, all of the threonines. Yeah, and then they just published it and didn't, and gave us the flies. It was awesome. So so these flies were available. We had to get them smuggle them in from China, but we got them, and unfortunately, the T11 alanine substitution homozygous fly is not viable. So we need to have a mother that lays these eggs. So we can't make a T11 homozygote, unfortunately. They die. They never live. But we can make a T11 heterozygote. So now half of the histone H3 in the embryo does not have T11, or has T11A, and they look like this. Their cell cycles are statistically significantly longer, and it's not on this graph, but occasionally we even see them do extra divisions. Or sorry, stop early, stop full cell cycle early. I think they are, yeah. So
the homozygous mutants are embryonic lethal, is it there?
Yes, they are embryonic or larval lethal. We never looked, and they reported that they don't come out. So I don't know when they die, but they don't produce mothers that we can use. So when we mutate T11, now we get the reverse phenotype. Cell cycles are longer. check1 probably can't interact as high affinity with these guys, and sometimes they even stop one cell cycle early, as predicted.
So you predict that heterozygotes for check 1 should have effects on cell cycle?
Yeah, maybe. So
Once a competitive substrate, there's a total activity of check 1.
right, but that assumes that check 1 total activity is dependent on check 1 abundance, and check 1 itself is phosphorylated to activate it, right? So check 1 is phosphorylated by ATR, ATM, and so this is me explaining why there's no phenotype.
There is no
There is no phenotype. No one's looked super, super carefully, but there's no obvious phenotype. They certainly don't like stop early or anything, or sorry, go extra or anything like that with any regularity. And so my hand-waving explanation, and this is hand-waving, is that check 1 is phosphorylated by its upstream kinase. And so the total pool of activated check 1 may not be directly proportional to the entire amount of check 1, right? Like if there's less, maybe the same amount or close to the same amount gets phosphorylated anyway by its upstream kinase, maybe. But yes, you're right, this would predict that.
above the Km of the kinases and phosphatases.
Right, that's hand-waving. These are the sorts of things that we would really like to get into, is like, what are the KDs and behaviors of all of these proteins, and abundances. The check 1 doesn't show up by mass spec, so I don't know what the physiological concentration of check 1 is. Yeah.
I'm just looking back at what I wrote
Yeah.
earlier. You said that you mutated a check 1 in a diploid and the cell cycle was its fastest in the
We-
was that?
So we made a check 1 double mutant, check 1 homozygous mutant, sorry. And those guys undergo faster cell cycles. In fact, they go so fast that at the end of 13, they're not quite done replicating their DNA when they enter mitosis, and they tear their chromatin apart. So check 1 homozygotes definitely have faster cell cycles. Check 1 hets, not obviously so. We should go back and look. We're also interested in looking at the check 1 hets, what they do in the context of the histone mutations, the histone tail manipulations. But yeah. Okay. So that's about all the data that I want to tell you. So I just want to sort of show this model. I think I've elucidated it. That our idea right now is that check 1 is trying to phosphorylate its target CDC25 in the early embryo, but it keeps bumping into all of these non-DNA bound histones that are floating around, and but rarely ends up finding its target and is unable to stop the cell cycle. Then, as development progresses, the increasing number of nuclei titrate out the histones. Now check 1 is able to find its targets, and now it's able to stop the cell cycle. So just to update our model for one last time, I'm going to move histones away from just affecting transcription to affecting both transcription and cell cycle directly. I'm going to add the specific model of how histones affect the cell cycle through inhibition of check 1. And yeah, that's...
So you think that inhibition of check 1 is physiologically necessary by that, because these are little mutations, right? If you mutate T11,
yeah. Right, so
homozygous, then
they're dead.
They're dead. So meaning that this inhibition is crucial for whatever, I don't know.
Yeah, and so
If it's only inhibition.
Right, so I don't know when or why they die, and that would be a really interesting thing to look at. But yeah, I think that this is important for cell cycle slowing, and if you don't slow your cell cycles right.
And this renin is conserved among species, right?
This what?
The T11 is conserved among
Yes,
species.
yes, yeah. Yeah, that's a conserved residue.
This
Yeah.
might be a really naive question.
Yeah.
Is check 1 interacting with DNA-bound histones as well?
Yeah, so in vitro, it can. I have not done the in vitro assays to figure out if the affinities are different. And so you're right. I sort of draw this model as, check 1 can interact with both, and the total amount is changing. But it could be that the total amount per nucleus is changing, or it could be that the relative affinities are different, or it could be some combination of those two. And these are all. This idea is less than a year old, and the experiments have been working beautifully, but there's so much left to explore.
And then just a quick follow-up. Could check one interact? Does it interact with H3.3? Like, that
The
was the one that's replacing, or,
right,
like,
so-
potentially replacing.
Right, so the T11's there, and in fact, H3.3 has another potential, it's never been shown, but potential check one binding site. So that could be interesting. And so in Xenopus, not by us, but by others, it's been shown that that H3.3 is important right after gastrulation, but in Xenopus, they can't do maternal effect mutants very easily, and so they can't do the experiment to ask what's going on with H3.3 requirements early. And so, yeah, these are all things that we're interested in. H3.3 may actually be a better inhibitor, but H3.3 is also in the nucleus early, so.
So if you overexpress check one, I mean, if this model is correct, overexpression of check one will alleviate the effect, right?
Again, it depends. It depends on how that affects the pool of active check one, right? So we would. Right, check one itself has to be activated, and so we would need to understand, like naively, yes, but.
But usually it's, I would be very surprised that, I mean, if it goes to, what you are saying that it goes to plateau, phosphorylated, I mean, you increase concentration, but phosphorylated goes to plateau.
Right, we don't know. We don't, right, we don't know how changing the overall pool size of the check one affects the active pool size of check one.
Meaning that the
The
rate
phosphor.
limiting is somewhere outside of check one, right?
For the pool size of check
But-
one. Yeah, maybe. These are questions.
Yeah.
So naively overexpressing check one, I would expect to have cell cycle effects. And I don't think anyone's done that. It would be a good experiment for us to do. But if it didn't, I could explain it away, I guess.
But you could always express a phosphomimicking mutant.
Yeah, right. So there is a phosphomimetic.
Well, not only overexpressing check one because it can do many things, but in conjunction with mutation T111A,
Right,
kind of to see whether
Right.
overexpression kind of
right. So we have,
rescues.
right, we have the crosses going. So overexpressing it, we just don't have the constructs yet to do. Knocking it down or making heterozygote, that's easy, like we have the flies. And so we have the crosses going to look at what happens if we overexpress the tails, both the T11A and the wild type tail, in the context of the het, of one copy of check one, because that's an easy experiment to do. I will let you know what we see. But yeah, these are all
Yeah,
good
it's
experiments.
Potentially possible, yeah, that plateaus, but I don't.
Yeah.
So it makes sense to me why, like, you know, every cell division you like open your nucleus and histones leak out, and then the daughters have less. But is it because H3 stops being made at some point?
So
Like it can't compensate, or?
Right, so a little bit of H3 is translated from maternal mRNAs, but the vast bulk of H3 is loaded as protein from the mother. So the total pool size is not quite constant, but close enough to constant that I don't worry about it.
I see. If you just take the whole embryo at
Right.
different phases and just grab it. Okay.
Right, so like there's this pool, like I drew with the purple thing at the beginning, right? There's this pool and you're running out of it. A little bit is made, so it's not the perfect simplest case, but it's less than double, and every cell cycle you double the number of nuclei, so it wouldn't get you that far.
I have a slightly different question.
Yeah.
I was thinking, do you have any idea if the concentration of histones in the cytoplasm might be affecting the cytoplasm biochemically, and then might be affecting the chromatin structure, compaction, and accessibility, and maybe that might lead to cell cycle changes, like maybe an indirect effect, or have you thought or explored that at all?
So what are you thinking? You're thinking, like flesh out what the model is.
I don't have a model.
Okay.
I'm just thinking that if you have more histones, are you going to get more compaction of the DNA or less compaction of DNA? Is it going to affect it? If it's affecting the compaction or accessibility, it might be affecting transcription, in addition to any other processes happening at that time, including, you know, dosage compensation, including pairing of the homologs
Yep.
in Drosophila and other processes.
Right. So not done by us, but done by Shelby Blythe, who's a new professor at Northwestern. He did a tax-seq over these early embryos, and globally, the number of nucleosomes doesn't change that much, he doesn't think. Specifically, at certain TSSs, they open up when they become transcribed. It's kind of hard to say if that's causal or correlate, you know, like upstream or downstream. Yeah, so this was sort of our initial model, was that, like, you know, histones are on chromatin. It took us forever to think of looking at histones not on DNA. Our initial model was going to be histones are going to be binding, you know, just super abundantly, and they're going to be blocking transcriptional start sites, and then you can't get transcription because there's just so many of them, and then you kind of start to run out, and now they can be moved around. I still think that that may be part of this, and that some loci may be more sensitive to histone concentrations because of whatever it is about their cis-regulatory regions. But I also think that you know, this tail isn't binding to chromatin. It's definitely not getting incorporated into nucleosomes, and it's having cell cycle effects. So that tells me that there's a role for histones that isn't in transcription. So it's definitely a thing we want to figure out, is how histones are affecting transcription, because in our biochemical assays and our best time-controlled time course RNA-seq, we think histones are directly affecting transcription. So yeah, and the obvious model is that its nucleosome occupancy is changing. Okay, I'll go through future directions really quick, if you guys don't mind. So yeah, the things that we're interested in are fully modeling the effects of histones on the cell cycle. We want to figure out all of the parameters. Figure out if there are other roles, either in the same role in other tissues, or just other residues that are important on histones off of chromatin, and then understand how changing histone availability affects the chromatin. And then in the longer term, I want to eventually move out of the embryo and into other tissues to look not just at cell size, but also at organ size, which can be fundamentally controlled in one of two ways. Either you control the cell size in an organ with a fixed number of cells, as is the case in the Drosophila oocyte, or you control the number of cells in a relatively fixed cell size organ, as is the case in the Drosophila wing disc. And so those are the places that I am headed in the future. And then I put the people who did the work's face on all of the slides where they did it, but just thanks to my lab, the wonderful fly community and frog community and microscopy community at Princeton, my collaborators at Penn, and then my former mentors at Stanford. And thank you guys. Any more questions? Yeah?
So the other genes that are just on some kind of timer, is that because you've got some maternal repressor that sometimes gets degraded?
Yeah. All right. So time-dependent genes. There's a lot that can be going on there. So there could be a maternal repressor that's getting degraded. There's also known to be activators, in particular the pioneer transcription factor ZELDA, that's getting translated. So they could be particularly responsive to the levels of ZELDA. Or they could be, I'm having a hard time distinguishing between things that are on a timer and things that are responsive to cell cycle time. And those are slightly different things, right? Like one is like when you got fertilized to time now, and the other is how much time have you just had to accumulate. And yeah, so all of those are open possibilities. And I definitely think accumulation of ZELDA is going to be important for a lot of genes. So that's definitely part of that. Yeah.
Are there other modifications on the tails that you know of, and is there like a way to measure them?
Oh yeah, the tails are very modified. I couldn't think of a good way of saying that. They're super modified. Yeah, like I don't know if you've heard, like all of these H3K9, H3K27, those guys are acetylated and methylated. There's tons of modifications on the tails. And so as we've been thinking about and realizing the importance of histones off of DNA, we've been wondering about how normally histones are not found in abundance off of DNA in like a normal cell cycle. They're degraded outside of S phase. But they are often overexpressed in cancers. And maybe there are other developmental contexts where they might be important, where the non-chromatin bound pools might be important. So yeah, we've been sort of excited about all of the possible things that could be going on with the other residues that could be being modified. but no evidence yet, one way or the other.
Okay, thank you.
Thank you, guys.